So good morning, everybody, and welcome to the the first of the webinar series, STEM in Healthcare. This is the first of 4 webinars that are going to be held this year jointly between the School of Mathematics, Computer Science and Engineering and the School of Healthcare Sciences at City University of London and the Institute of Physics and Engineering in Medicine. My name's Stephen O'Connor, and currently I'm the president of IPEM. And it's my pleasure to chair this webinar. So at any time you can submit questions on the using the Q&A box throughout, and then we'll come back to those at the end of the session to have questions. So what I'd like to do is to introduce Professor Panicos Kyriakou, Director of the Research Centre for Biomedical Engineering at City, whose talk is on optical sensors in traumatic brain injury. Panicos, thank you.
Thank you, Stephen, and good morning to everybody. It is really a great pleasure for me to be able to participate in this webinar organized by probably the 2 institutions that are closest to my heart. There is my university, obviously City, and IPEM, where I have been a member for decades. So it is a true honor, and thank you for inviting me. As Stephen said, I will speak and focus the discussion on the optical sensing for traumatic brain injury. I'm just gonna go through very selfishly a couple of slides on the center at City, where colleagues joining us today, they can perhaps, as in many meetings, follow up with discussions in collaborative discussions. So we are a center where we are focusing in the development of sensors, optical sensors primarily. A lot of wearable technologies in order to be able to develop technologies for the non-invasive early detection of cardiovascular disease. And for the last 25 years or so, we've been focusing in developing sensors for non-invasive monitoring of blood and tissue absorbers or biochemistry. And a small group within the center also is focusing in neurodegenerative conditions, applying sensor technologies. So here you can see some of the main highlights or areas of strength within the center, which is sensors, wearables, optical and impedance spectroscopy, neural interfacing and neuroprosthesis. Of course, with any technology, you need a lot of signal processing. And in the last few years, amongst many, we have strengthened the center with expertise in machine learning and AI in the biosignal analysis as well. We are a decent sized group with a group of academics, many collaborators, especially clinicians, supporting the clinical aspects of the projects that we run. We are lucky to have a bunch of industrial collaborators funding some of our work. And of course, the work cannot be done by not having a strong group of postdocs and PhDs. And my thanks go to all of them. Without their contribution, we wouldn't be able to accomplish what we have accomplished. Within the team, we are very techies. We love building things. We have a lot of curiosity within the center. So we design and fabricate pretty much everything in-house within our laboratories. We develop optical sensors for different applications. We have been working for many, many years with the NHS, primarily BART, the Royal London and Great Ormond Street, our main partners in developing sensors, which can be transcutaneous, completely non-invasive, or they can be more customized. They can be indwelling, whether esophageally or rectally, or sensors for in splanchnic sensors, for example. Of course, we develop all the associate software to be able to provide to the clinicians the signals, both in a qualifying manner and in a quantifiable manner, so they can see both the physiological signals and the results, whether it could be oxygenation, heart rate, or whatever else we are investigating in that particular project. Within the center, we have facilities and resources to conduct in vivo studies on volunteers within the remit, obviously, or the ethical limits. And following the in vivo studies within our physiological laboratories, we will take the technology to the hospitals and proceed with clinical trials. In many projects, we have to conduct in vitro work, whether it's wet work or modeling work, or phantom work in order to control the experiments depending on what we are trying to investigate. Our group, proudly, is probably one of the biggest photoplethysmography group in the world. We are one of those groups that it does both applied and basic research in photoplethysmography. We are investigating the origin of the signal and its applications and its relations to hemodynamics, vascular mechanics, and hemorrheology. And it gives me great pleasure today. I've used this platform to announce for the first time that a book is coming out, which is edited by myself and John Allen, on photoplethysmography. So it will be the first comprehensive book on PBG. And of course, our applied research takes us to many applications, and some of those you can see them in this list, which is not exhaustive. So amongst many other groups around the globe, we are also exercised with using optical technologies and PBG in order to apply these to healthcare technologies and different pathologies or pathophysiological phenomena. We do a lot of signal processing and a lot of machine learning, a lot of feature extraction from the biosignals, which is currently one of the trends in people developing smarter wearable technologies. We do a lot of modeling of light tissue interaction. It helps us in designing the sensors. So depending on the application, we can develop very comprehensive Monte Carlo simulations in order to understand the behavior of light in tissue at the different layers. I'm going to now get into the subject of the study, which is traumatic brain injury, and one of the primary biomarkers or the gold standard of measuring intracranial pressure for assessing TBI is the measurement of intracranial pressure. So a little bit of background on traumatic brain injury, and a lot of people, they name it as the silent epidemic. And you can see that a huge volume of people are affected by traumatic brain injury. And, this can come from everywhere. You have accidents, whether it's car accidents, you have work accidents. It could be in sports. It could be in people working in harsh environments. It could be the military. So the volume of people affected by traumatic brain injury, it is very large. The mortality is very high. And the financial burden of TBI across the globe exceeds $400 billion annually. So, and bringing into home within the UK, we have over 1 million people living with long-term disabilities or traumatic brain injury. So it's a pretty devastating pathology or problem. Now, if you're a neurocritical care expert or a neurosurgeon, the gold standard of assessing TBI is to measure the intracranial pressure. You have an accident, you get a blow on the head, your intracranial pressure rises, and then obviously it creates swelling, it can create bleeding, hematomas. And the neurocritical surgeons, they need to decide when to measure the intracranial pressure. And the reason for that decision that makes it very challenging, it is because it is so invasive. It is one of the most invasive non-therapeutic procedures. Basically, the neurosurgeons, they have to drill a hole in the skull and insert a pressure transducer within the brain, literally stabbing the brain with a pressure transducer. So it is extremely invasive, and therefore they have to be very selective in who they will put a bolt. They cannot just put a bolt into nilly-willy into anybody who walks into neurocritical care. And of course, we cannot put a bolt on a patient that he just had an accident on the side of the road when the ambulance arrives. So that access is not possible. So it's called the golden hour. We don't know what happens to somebody's intracranial pressure until it reaches a hospital. In a neurocritical care unit, they will assess the patient first, and then they will decide whether they will need to put a bolt to measure the intracranial pressure, the gold standard for a traumatic brain injury. And obviously, apart from the invasiveness, it comes with a lot of risks. You can have hemorrhage, you can have cerebrospinal fluid leakage, it can lead to infection. So there is a lot of concern how we apply this invasive procedure to the patients. And of course, TBI, it overspills to a lot of communities. You have severe TBI, where we put a bolt in those patients most of the time, but we have a lot of mild TBI and a lot of moderate TBI, which actually neurosurgeons, they don't put a bolt in this, a transducer in these patients. We have traumatic brain injury and or increases in intracranial pressure in patients where they suffer with migraine, with stroke, with meningitis, with hydrocephalus. And therefore, it would be great to be able to assess their intracranial pressure in a non-invasive manner. The vision, the motivation for us biomedical engineers, as it is always, we are trying to find the limitations of current technology or the gaps where technology does not exist, and then go back into the drawing board and trying to come forward with technologies to solve the problem. So for us, the vision is to develop a non-invasive ICP monitor to replace this most invasive monitor in clinical practice. So we have been working on this NIHR-funded project for the last 4 years in collaboration with the Royal London Hospital, one of the biggest neurocritical care units in the land. Obviously, being very strong in photonics and optics and biomedical optics, we're using the principle of light diffusion in tissues. So we do know that certain light, certain wavelengths can penetrate within the scalp and allow us access into the brain. And we can develop technologies to be able to interrogate the brain. So using our skills and expertise, we have developed a reflectance optical sensor comprising of 4 wavelengths and 2 photodiodes, a proximal photodiode and a distal photodiode, allowing the interrogation of proximal signals, so optical signals or PBGs, before they enter on the brain, basically from your foreheads. And then we have a distal photodiode that it will penetrate light into the brain and bring optical signals back, representing changes, volumetric changes in the brain. So it is a pill and stick technology, and you can see this in one of our volunteers, completely non-invasive, very lightweight, and the technology at the moment is connected into a processing system. The slide here shows for the first time some typical optical signals. I will be referring them as PBGs, as photoplethysmographs, from the proximal and from the distal sensor. So we have PBGs at 850 nanometers, 880 nanometers, 810, and 7070, all of them wavelengths within the near infrared region of the spectrum. And again, these are PBGs coming straight from the distal, which is from the depths of the brain. So high quality signals are all wavelengths, which brings a lot of confidence that we can interrogate the brain and we can bring signals back, and which can give us information of what's happening within the hemodynamics of the brain. Of course, you can develop the best instrumentation, but you need those signal processing techniques to be able to squeeze the signal and get the information out. Our motivation, our primary motivation, is to use this optical signal, this PBG, and be able to identify features within the PBG that they relate or correlate with changes in intracranial pressure in this non-invasive manner. And you know, people in the field, especially now with machine learning, they're using many features from the morphology of the signal. And for us, we made initially almost like a short list of signals. So of features, we have the pulse amplitude, the upstroke of the signal, the rise time, the early diastolic decay rate, the pulse width, the late diastolic area under the curve, etc. You know, a lot of people, they can extract up to 50 features from the PBG. But for us, that was the starting point to see if any of those features, they have a close link or a relation with changes with intracranial pressure. We have developed a regression model, and I have to confess, I'm not a machine learning expert. So the team is a collaborative project, have developed the model to be able to identify the features which they relate to changes in ICP. So the first step, as we are all, good practice, we develop an in vitro evaluation, a control environment within the lab in order to see how this will work. So we develop a phantom, a brain phantom, that actually we can change the intracranial pressure within the phantom. We created a model which is perfusing the phantom. We have a pump that is circulating blood or blood analogous, and we developed a sensor on top of the phantom. So this is schematically this control environment to allows us to make some preliminary experiments. We have the pump, we have the vasculature branching from the carotid, supplying the aortic branch, goes into the brain. So we have the scalp and the phantom inside. The phantom inside the scalp is vascularized. So we are pumping fluid or blood analogous within the pump, within the phantom, and on top of the phantom, we have the sensor. And this is a photo of the setup. This is the scalp. I know it doesn't look like a scalp, but volumetrically, the dimensions or the volume of this compartment, which we can compress, we can induce changes in pressure, is similar to the human adult scalp, and the optical sensor on top. The first experiment we did, we have increased the intracranial pressure within the compartment, within the scalp, and we are looking at PBGs at each step of the change in intracranial pressure, starting from low 10 millimeters of mercury up to 40. Of course, the patient at 40 is probably dead. So we're looking, these are the typical intracranial pressures that you would see in a patient. And if it rises above 25, that is very dangerous. So we can see clearly a beautiful step change of the optical signals at every control change in intracranial pressure, which gave us a green light that actually we can observe PBG signals at different intracranial pressures. And then we've looked at all the features and see what are the features that actually prominently change with changes in ICP. And I will only shortlist two. Look at the amplitude of the PBG as we increase the intracranial pressure in this in vitro model. You know, pretty steady state, but when we increase the pressure about 15 millimeters of mercury, we have this significant drop in the amplitude of the signal. The same with the upslope, you know, pretty steady state. When we reach the 14, 15, which is a critical ICP pressure for patients, we have this drop again. So for us, we said, wow, great. These two features are really responsive to changes in intracranial pressure. And we made this analysis for every feature we explored. And therefore, in summary, this in vitro model gave us the first green light. We can predict using our model accurately the intracranial pressure from the in vitro model, because in the in vitro model, we know exactly, we control the intracranial pressure. And look at the results, the correlation, the RMSE, the limits of agreement, too good to be true, to be honest. So following this, we went into a clinical trial. We did a small study on volunteers. I'm not presenting here due to time. And on volunteers, of course, you cannot put an intracranial bolt in their head, but we did some tilting where we induce engorgement of the brain by tilting them, standing up, and we can see the changes within the brain. But obviously the proof of the pudding was in the clinical trial. So here we have a patient where this patient has our sensor on the forehead taped down. The patient has an invasive intracranial bolt, which is the gold standard. All the clinical trials are at the Royal London in the neurocritical care unit. We had some on and off interruptions due to COVID, as many people suffered during with their clinical trials. So far, we have performed 30 studies in 30 patients. The project is funded for 40, and we have evaluated offline our model. So these are some results from a group of nine patients. Now we have more results. And again, you can see a Blunden-Ullman here with the limits of agreement on this preliminary pilot study. And again, in summary below, you can see the correlation, the root mean square error, the limits of agreement, the ROC results, the sensitivity at 20 millimeters of mercury, and the specificity. I mean, these are amazing results as a first go, which suggests that the way we are approaching it could end up producing a completely non-invasive intracranial pressure technology. So closing this presentation, very proudly, this project was presented in the STEM for Britain Awards in 2020 in the House of Lords, and we have received the gold medal. Thomas, which was the research fellow on the project, was there to receive it, and it was a very proud moment for us, but also a moment that demonstrates that this is such an area of need which has been recognized during that event. So with that, I want to thank you very much, and I will be very happy to receive any questions. But also I will invite anybody from the audience, if they wish to visit us and open a dialogue, it will be great. Research can only be done through collaboration. So thank you very much for your time.
Thank you very much, Panicos. A fascinating experience looking at the comparisons with the gold standard and your own developments. Fascinating. So thank you. So I would ask Professor Tong Sun, who's a Professor of Sensor Engineering at City University of London, please to make her presentation, Optical Fiber-Based Chemical Sensor Technologies. Over to you, Tong. Thank you.
Thank you, Chairman, for your very kind introduction and for the invitation. Good morning, ladies and gentlemen. The topic which I'm going to focus on today is on the optical fiber-based chemical sensor technologies, which we have explored over the past number of years for different industry applications. I do hope that some of these technologies could possibly be used in healthcare. The work which I'm going to present today is a joint work with my colleagues. Dr. Nguyen is a chemist, Drs. Fabian Vidovic and Ranti are engineers, and Professor Grattan is a physicist. Then you can tell this is a multidisciplinary team for the sensor development. To start with, I would like to share some of the key features that optical fiber sensors can offer, and which are unrivaled by their electrical counterparts. The first one is their immunity to EM interference, which means they can be used in those environments where EM interference is a big issue. For example, for MRI scanning, for those with a high current, high voltage conditions, or microwave conditions. The fiber is of a small size. The diameter of fiber is only 125 micron. So they are easily embedded into the structures or materials to make the structure or material to be smart. Fiber is based upon the telecom fiber, so unavoidably they can be nicely used for long distance measurement with minimal loss. Again, the fiber is of silica, which means they are chemically inert, so they are highly suitable for harsh environmental workings. For those, for example, for those corrosive, explosive, and underwater and radiation conditions, or those can be even sterilized for the medical use if needed. If we are using some of the spatial sensing techniques, for example, fiber backgrading technique, then the measurement could be multiplexed and making it to be quasi-distributed, aiming for multi-point and multi-parameter to be measured simultaneously. Before I move on to talk about the first chemical sensor design, I want to share with you of this video, which we downloaded from FBGS. This video clip nicely describes the sensing principle of fiber gratings. So let me actually run it now.
Fiber-Bragg grating sensor principle. Fiber-Bragg gratings, in short FBGs, are an elegant breed of intrinsic sensors, which are deployed along the length of an optical fiber. The individual FBGs is fabricated by inscribing an invisible permanent periodic refractive index change in the core of the optical fiber. When an incident spectrum of light propagates through the grating, a specific wavelength, named the Bragg wavelength, is reflected back, while the rest of the spectrum is transmitted unaffected. When an external axial strain is induced, the FBG reacts accordingly, causing a proportional shift in the reflected Bragg wavelength. With a one-time instrument calibration, Strain values and many other derived parameters can be measured dynamically. A key advantage of FBG technology is that measurement points can be fabricated as an array of independent sensors along the same fiber, enabling multiplexed or even distributed measurements. These multiplexed FBG sensors can also be used to monitor other physical parameters, such as temperature, in real time. Contact us today to discuss your application.
As described in the video clip, fiber Bragg grating is sensitive to both strain and temperature. Now, when we are using this fiber Bragg grating for the human sensor design, FBG is used as a sensor substrate. So to make it as a humidity sensor, what we did is to coat a thin layer of polymer on the surface of fiber Bragg grating. And the approach you can use, for example, to use a dip coating machine to allow this fiber Bragg grating to be coated. Now, once it's coated with polymer, polymer responds to the humidity level in the surrounding environment. And then with the increase of the humidity level, then the polymer swells and then induces strain onto the fiber Bragg grating it is coated on. So in this way, there's an obvious wavelength shift. And then the IH level is increased, the shift is higher. So this is based upon the strain effect of the fiber Bragg grating. So if we summarize using equation, you can tell the wavelength shift of the grating itself now is formed by two key components. The first component is coming from the strain induced by the humidity level. The second component is coming from the temperature effect, either through this thermo-optical effect or through thermo-induced strain effect. But to separate these two elements apart, then we need to consider all of these in the sensor design. So one typical design I have used is shown in this diagram here. Instead of using one grating, we are using two gratings, and FBG1 is coated, so it is sensitive to temperature only. And the second one, FBG2, is actually coated. Then it is sensitive to both humidity and strain. Now we are having two channels information and have two unknowns. So using this approach, then we can separate easily the temperature it is and what the IH it is. Of course, after actually its manufacturing, we need to make sure the sensors are properly packaged for different industry applications. And this is one type of design, package design we used for humidity monitoring in sewers. So this comes down to the application of how the humidity sensors being used in the real world. What I'm going to show you here is only two scenarios and joint work with Sydney Water in Australia. So that's for the sewer network condition monitoring. The diagram below here shows the sensor installation locations at two sites in Sydney, and the right-hand side shows actually how the sensors are being located. This allows us to continuously monitor or measure the humidity in this highly acidic environment. And the data have been used to help Sydney Water to establish models to determine end of the service life of sewers and identify where to carry out the sewer rehabilitation. So this is one deployment, and the sensors have been there for more than 2 years now, and they still function nicely. We continuously receive the data from them. Following this success, then we are able to actually install these sensors in biodigester, again, in collaboration with Sydney Water. The picture below here showing a layout of biodigesters and their control system and control unit and the control room where our monitoring interrogation system are installed. The sensors are used to monitor the humidity inside the tanks with high levels of methane, carbon dioxide, and hydrogen sulfide gases. Then you can imagine before the installation, we have gone through rigorous safety clearance before they are used in the North Head sewage treatment plant in Sydney, Australia. Because of difficulty to access the site, the sensors are exposed to the highly dangerous site, but data from the interrogators would be transmitted to the control room via the 4G network. So this is one type of sensor, which I'm going to talk about. And the second one I'd like to highlight is heavy metal sensors, which have been developed for inspection of contaminated soil, because heavy metal sensors are considered one of the major contaminants in some of the European countries. And then the project is funded by EU. But if we look at very closely the sensor design here, it is mainly formed by two components. The first one is a receptor. Now we used it as a chrome sensor as a receptor specifically for the mercury detection. Because we are using fiber as optical sensor substrate, and we need to have optical signal. So the optical signaling is realized by fluorophore, which is polymerizable coumarin. So once actually these materials being manufactured, they are immobilized onto the sensor substrate here. So this is the sensor itself. Sorry, so I keep on, because I'm using the mouse, so it's keep on. You can see actually for this diagram, this is the sensor layout. We have the receptor, we have the fluor, we have the fiber, which is the sensor material is immobilized into. However, if you look at the first diagram here, when the receptor empty is energy level like this, and then the fluorophore, the energy level is laid out like this. But when there's no presence of mercury, which means the receptor currently is empty, then when it is pumped, then there's obvious photo-induced energy transfer on receptor down to the fluorophore. So that kills the fluorescence. In this case, when there's no presence of mercury, and then there's no fluorescent signal. However, if we move down to the second case, when the mercury ion is being present, then the receptor binds that and then make this to be occupied. But once it's occupied, its energy level being changed. And in this way, the initial photo-induced photon electron transfer, now that part has been cut. Then excited electrons in the fluorophore material can be easily dropped to the ground level, and in the mean release the photon to create the fluorescence signal. So the signaling is straightforward. When there's no concentration for high mercury being present, there's no fluorescent signal. But when there's actually the presence of mercury ions, then there's triggered the fluorescent signal. With more being bound by the receptors, the signal is getting stronger. So this is the basic sensing principle for fluorescence-based mercury sensor design. Its manufacturing, which Dr. Hian Gwen has done, is first of all to functionize the surface of the fiber itself, and in the meantime, preparing of those pre-polymerization mixture. Following this, she can either use the thermal polarization process or using UV polarization process, and that way would work. And in the end, the probe you can see is the one which is on the right. Then at the tip of it, that's the sensor material being coated onto the fiber. Now, in order to capture the signal, we need to create a sensor system. So the left-hand side is the sensor system, which is very, very simple. So first of all, we have 1x2 fiber coupler. At the end of it, there's a sensor probe. Here, it is a mercury sensor. In order to pump its signal, we need to have a light from LED. So LED light is coupled through one branch of the coupler, and the fluorescent signal generated from the sensor probe is channeled out by the other branch and measured by the mini spectrometer. So the spectrum you are looking at is you have both the excitation light source as well as the fluorescent signal. So the right diagram actually shows when the mercury concentration increases, the fluorescence signal increases as well. And in this case, we have checked the mercury response in the range from 0 to 28 micromole, and detection limit is 0.15 micromole. Now, the question is, this is designed for mercury detection. Does it sensitive to the other heavy metals? So it is important to have a check, it's a cross sensitivity or interference checking. Hian has mixed the solution with various type of metal ions and using this probe to do the measurement. Then left diagram shows this sensor specifically designed for mercury detection, shows the much higher sensitivity to the mercury only compared to those other metal ions. So it does show there's a high excellent selectivity for this sensor for mercury itself. So this is where the selectivity is required. Once you have used it for sensing, you want it actually to be repeatably can be used, which means we need to make sure the sensor is reversible. So here using agent EDTA to allow the sensor to be regenerated and then washing it after each use and then to make it start again. So in this way, the sensor can be used multiple times, and which means sensor is reversible. And the regeneration time here used is around 15 to 30 minutes. So the receptor, if I recap, for this mercury sensor design, it has a receptor specifically designed for heavy metal recognition, in particular for the mercury in this case, and the signaling is the fluorophore. But when we change the analyte, and the receptor need to be changed as well. Then Hien has used another approach to create the sensor. The approach she used is called the molecular imprinted polymer to create a receptor. Now, this is joint work with the Home Office and Smith Detections for drug detection. To make this such a sensor, we need to have a drug to be present, first of all, as a template. So you can see we purchased the cocaine from legal sources, of course, and then to allow the sensor manufacturing. So again, we have the template being purchased, being present, and we have the signaling, polymerizable fluorophore, and the fiber. And then we use a similar process, which I described earlier, to create this polymerization, through this polymerization process, create a complex like this. If we take out the cocaine, then this is the sensor which is being formed. Now, if I repeat again, the receptor now is this part, which has a shape and a size which is complementary to the targeted compound. In this case, is cocaine. And we have the fluorophore, which is used for signal, and the fiber is used for the signal transmission and for the signal collection itself. This is now the sensor probe, which is made available for detection of the cocaine. Once the cocaine is present, then it will trigger the fluorophore to react to create the fluorescent signal. So if you look at the probe itself, this is actually on the normal conditions when there's no presence of the cocaine, and then this actually shows that the cocaine is present, and then fluorescent signal is responding to the cocaine concentration. So this is the signal, the result which you have obtained by increasing the concentration of the cocaine and looking at its fluorescent signal change. It is noticed there's an increase of the intensity as a function of the concentration increase. However, it reaches saturation at around 500 micromole is because all the binding sites has been occupied, so there's no more signal to be shown. Both the signals, which I mentioned, that both the cocaine sensor, MIP cocaine fluorescence sensor, as well as the mercury sensor, we all use fluorophore as a signaling. But sometimes it may be difficult for the sensor design to conjugate the fluorophore into the receptor to facilitate that photo-induced electron transfer. If that is the case, then we may be able to replace the fluorophore, use the other signalings. One possibility is to use a surface plasma resonance approach. Then this is able to replace fluorophore and to generate optical signal. And then in this way, then you don't need to worry about actually the fluorophore, which is fluorophore you should choose to allow the clear matching. The difference is that the fiber is no longer just a simple fiber, but now we need to coat the fiber with a layer of gold to form this SPR, surface plasma effect, which is created by the conductive electrons oscillate collectively with a resonant frequency at the interface between the fiber and the gold interface. The receptor itself, making it is using a similar way I mentioned earlier. So if we combine both together, this is another type of sensor, but this is SPR-based MIP cocaine sensor. Again, with the presence of the and it will be locked into this receptor, induces the dielectric constant change surrounding the gold film. Then this will causes the signal, SPR signal wavelength shift, as shown on the right. Following this, we need to see its sensor system. Similar to the sensor system I described earlier for the fluorescence-based sensor, but the major difference is the light source. We no longer use a narrowband light, but replace it with a broadband light source. And in this way, we are able to track down the resonant SPR signal shift. And in this, we test it with the concentration of cocaine from 0 to 400 micromole. And it's noticeable there's obvious SPR wavelength shift to the left, decreasing wavelength shift with increased concentration. Then it does show that the sensor works when it responds to the cocaine, and then there's obvious signal change being captured by the system. The probe on the right is actually the SPR probe coated with gold, and again coated with a MIP receptor. So I have actually run through a few sensor designs, but it may be impossible for me to run through all the designs, but I just want to summarize briefly. The sensor designs could be diverse, but the receptor itself, we need to make sure it's specific target analytes. Here we have used the polymer for humidity. We have used the receptor, metal receptor for the mercury. We have used a MIP for drugs. And optical signaling could vary as well. We can use fluorophore to create a fluorescent signal, using FBGs to pick up the strain effect, using SPR to pick up the resonance of the change surrounding the dielectrical material and the gold film. Of course, we can use the other approaches, such as long period gratings, etc. But after all these sensors were made, then they need to be packaged properly before they are used in the field. So I would like to finish by showing you this picture. This is actually the picture of Hong Kong Zhuhai-Macau Bridge. This is where we have some of our chemical sensors have been installed down there for remote monitoring. So I'd better stop here, but I would like to thank you for your time. I'll be very happy to answer any questions you may have later on. Thank you.
Thank you very much indeed, Tong. So fascinating work there. Thank you. So the third presentation today is by Peter Charlton, who's a British Heart Foundation Research Fellow working at the School of Clinical Medicine in Cambridge. Peter is going to present Realising the Potential of Wearables for Health Monitoring. Over to you, Peter. Thank you.
Thank you very much. Just as I start, there are a couple of links in the chat which may be helpful. So hello, and thank you for the invitation to speak today. I'll be presenting on realising the potential of wearables for health monitoring. Cardiovascular disease accounts for over 1/4 of deaths in the UK. During this tour, vascular disease will cause 3 hospital admissions, 5 deaths, and cost the NHS £250,000. The growing use of wearables provides opportunity to assess cardiovascular health in daily life and potentially reduce the burden of cardiovascular disease. Indeed, it's expected that in 2 years, there will be over 1 billion wearables globally. Most wrist-worn wearables which monitor heart rate do so by measuring the arterial pulse wave, which contains a wealth of information on the heart and blood vessels. Potentially, the signal could be used not only for heart rate monitoring, but also to provide additional information, including the heart rhythm and other vital signs, such as blood pressure and respiratory rate. To introduce myself, my name is Peter Charlton. I'm a biomedical engineer based at the University of Cambridge and here at City. I work alongside clinical colleagues, such as at St. Thomas's Hospital shown here, to develop signal processing techniques for wearables, which could be used for clinical decision making. In this talk, I'll provide an introduction to using consumer wearables for health monitoring. I'll provide an overview of the development of wearables, then highlight potential clinical applications of wearables, demonstrating how they could be used to improve health outcomes. And finally, I'll highlight next steps to ensure wearables can be used to inform clinical decisions both safely and robustly. So, the development of wearables. The development of wearables has taken place in two arenas, clinical and consumer settings. Clinical wearables have been primarily focused on ECG monitoring to diagnose heart problems. Holter monitors entered production in the early 1960s, allowing ECG data to be recorded for retrospective review. By the late 1960s, data from wearables were transmitted from the Apollo missions back to Earth for near real-time review. This approach began being used on hospital wards in the 1980s for real-time ECG monitoring. Since then, some clinical wearables which monitor oxygen saturation have entered use. In contrast, consumer wearables have been primarily focused on activity tracking and heart rate monitoring. Also in the 1960s, pedometers for counting steps became available. Heart rate monitors became available in the late 1970s in the form of a chest belt. These are designed for ECG-based monitoring during exercise. In the early 2010s, wrist-worn devices became available, which could monitor heart rate at the wrist without the need for a chest belt. Looking back, the same sensing technologies have been used in both settings for different purposes. There are three key sensing technologies used here. Firstly, electrocardiogram monitoring, or ECG for short. The ECG is a measure of the electrical current generated by the heart each heartbeat. It is acquired by measuring the voltage difference between 2 points on the body surface over time. Traditionally, it's been measured on the chest, close to the heart. Recently, chest patch devices have been developed which measure the ECG between two electrodes in close proximity. In addition, wrist-worn devices have been developed which can measure the ECG between opposite arms. The ECG can be used to diagnose a range of heart conditions. Secondly, photoplethysmography, or PPG for short. The PPG is a measure of changes in blood volume over time in a bed of tissue. It's usually acquired by illuminating the skin with an LED and measuring the intensity of light either transmitted through or reflected from the skin. The PPG is widely measured by pulse oximeters at the finger, but many wearables now measure it at the wrist. The PPG is used in wearables primarily for heart rate monitoring, but can also be used to assess oxygen saturation and several other parameters. Thirdly, accelerometers are used to detect and quantify movements. Accelerometers measure acceleration and are widely incorporated into wearables as they are cheap and consume little power. Accelerometry can be used to count steps, identify falls, and as a reference motion signal with which to either identify or remove the effects of motion from other signals. The functionality of wearables is mainly determined by their sensor technologies and the algorithms used to derive parameters from the sensor signals. This table summarizes the functionality of a few selected consumer wearables. Most of these wearables use the PPG for heart rate monitoring, but fewer identify an irregular pulse. Recently, many wearables have incorporated oxygen saturation monitoring. Other PPG-derived parameters, such as respiratory rate and blood pressure, are not yet commonly provided. Simultaneous PPG and accelerometry signals can be used together to assess sleep patterns and parameters related to exercise and fitness. All the wearables considered provide a step count from the accelerometry signal, and some wearables have additional sensors, such as an ECG sensor, a barometer to measure elevation, which can be used to identify stair climbing, and perhaps a thermometer, providing temperature. This table demonstrates the wide functionality of consumer wearables. Since consumer wearables are becoming more widely used, there's potentially opportunity to use their data for clinical decision making. So, how could consumer wearables be used to inform clinical decisions? Atrial fibrillation is the most common abnormal heart rhythm and is associated with a 5-fold increase in stroke risk. Fortunately, if it's recognised, it can be treated with medication. However, AF is often not recognised as it can occur only intermittently and without symptoms. Consequently, detecting AF is an ideal clinical application of wearables. Wearables could provide added monitoring to detect an otherwise underdiagnosed disease, which can be treated to hopefully improve health outcomes. Atrial fibrillation can be identified from the PPG signal by assessing the regularity of beat-to-beat intervals. In a normal rhythm, the beat-to-beat intervals are fairly consistent. In contrast, in atrial fibrillation, the beat-to-beat intervals are irregular. Potentially, a consumer wearable could be used to identify possible AF episodes in this manner. Indeed, the potential utility of an Apple Watch to identify AF was recently assessed in the Apple Heart Study in over 400,000 individuals. Key results included a reassuringly low alert rate, a high positive predictive value of alerts, and a much longer monitoring time than could be achieved at scale in clinical practice. In this study, possible AF episodes prompted further monitoring using the gold standard of an ECG-based wearable to confirm a diagnosis. The recent addition of ECG sensors to wrist-worn wearables means that potentially they could be used to not only identify possible AF, but also to prompt the user to take an ECG measurement, which could be clinically reviewed to confirm a diagnosis. As a second case study, infectious diseases such as influenza and now COVID-19 have a profound impact on morbidity and mortality. Information on the spread of infectious diseases can be used to direct healthcare resources, plan public health interventions, and potentially even provide early warning to individuals that they may be infected. Recently, the potential benefits of using Fitbit data to track influenza-like illnesses were assessed, using at least 60 days of data from almost 50,000 people in the US. An elevated resting heart rate and increased sleep duration were used as markers of disease. The study found that the inclusion of Fitbit data improved the performance of disease prediction models, which could help ensure timely responses to reduce further transmission. The potential benefits of wearables for identifying COVID-19 have also been assessed. The inclusion of data from consumer wearables in COVID-19 detection models was found to improve performance beyond that of using symptom app data alone. In addition, data from smartwatches has been found to aid pre-symptomatic detection of COVID-19. Several parameters were used in these studies: the resting heart rate, sleep duration, step count, and the heart rate to steps ratio. In the future, early warning systems could integrate wearable data, self-reported symptoms, and electronic health record data, providing both population level and individual level surveillance. I've described a couple of exciting applications of wearables for health monitoring. Time would fail me if I were to describe all of the potential applications, so here is a list of applications to look out for in the future, and a couple of references to publications currently under review, which describe them in further depth. So, what needs to be done to realise the potential of wearables for health monitoring? In my view, there are a series of next steps. Firstly, wearable devices will continue to be optimised to provide the best possible signals for analysis. Recent innovations include using different wavelength LEDs during different activities, as PPG signals acquired at different wavelengths have different strengths and weaknesses, and using multiple LEDs in a single sensor to acquire multiple PPG signals with which to improve the signal-to-noise ratio of a final composite PPG signal. Secondly, signal processing algorithms must be robust to provide accurate and precise parameters. training datasets are invaluable for developing algorithms and should contain wearable sensor signals alongside reliable reference parameters measured in the target population and the target setting. Thirdly, analytical techniques must provide clinically useful information. For instance, in the Apple Heart study, notifications of an irregular pulse were only raised if at least 5 out of 6 consecutive measurements exhibited an irregular pulse, thereby reducing the false alert rate. Fourthly, wearables need to be tailored towards the target users, particularly as those who may benefit most from wearables are not necessarily those who currently use wearables most. We need to understand what motivates people to use wearables and ensure they are designed accordingly. Finally, and most importantly, specific clinical applications should be identified in which wearables could contribute to improved patient outcomes. Usually, such applications involve either identifying an under-recognized disease or providing monitoring in a setting where it would otherwise be impractical. Here are some helpful resources accompanying this presentation, including the slides. The links are in the chat. At this website, you'll find datasets which I've used in research into wearables, signal processing algorithms, presentations, including this one, and publications. I'd like to thank you for listening and thank my colleagues for their valuable input and guidance as we've been developing our understanding of wearables, and my funder, the British Heart Foundation, without whom this work would not have been possible. I'm always pleased to hear from others. Do feel free to get in touch. To conclude, consumer wearables now have a wide functionality. Clinical applications are emerging, including detecting atrial fibrillation. And when used clinically, wearables should be like a harness. They should be highly reliable and for specified purposes. Thank you very much.
Thank you very much, Peter. Very nice presentation indeed, sort of covering the wearables market. Thank you. There are some questions that are on the Q&A session. So David Delpy, would you like to ask the question that you have for Panicos, please?
Yeah, sorry, am I unmuted now?
Yes, you're fine. I can hear you. Thank you.
Right. Well, I had 2 questions, one for Panicos and one for Peter. But since Peter's just finished speaking, can I ask him the one I asked particularly for him, which is that a common problem with wearable signals, and especially the optical-based signals that you're talking about, is the presence of skin coloration and high melanin absorption in some patient groups. Is that going to be, or continuing to be, a major limitation in terms of wearables for certain clinical groups?
Thanks, David. It's a very important question. So I was looking into the literature on the impact of skin color on the accuracy of parameters obtained from wearables, and I didn't find an awful lot of evidence on it. And the evidence that I did find suggested that heart rate measurements were not affected by skin color for a particular device, and that there was some impact of skin color on PPG measurements, but that it wasn't necessarily prohibitive to obtaining accurate parameters. I think we need to do further research in this area. I think it's an important question to always keep in mind. But at the moment, fortunately, the wearables for which there is evidence that I'm aware of don't exhibit large differences between different skin colors.
Yeah, no, I mean, measuring a pulse, the pulse itself is not affected by, but the size of the signal that you get, and hence the proportion of the time for which the signal is acceptably good or good enough to be analyzed is the problem. Usually, it's attenuation of the signal rather than the actual presence or absence of the signal.
Well, it would be great to be aware of the evidence behind that. I'd be fascinated to read more, David.
Okay, there's a lot of stuff on the pulse oximetry sites which relate essentially to a PPG type signal.
Thank you.
You want to go on with your question to Panikos, David?
Okay, right. I wasn't sure if I was allowed to. Okay, in that case, Panikos, it's a similar sort of question, which is that in head injury and patients in ITU, a major problem is for optical measurement is continued slow bleeding, either into the brain or the presence of blood in the or small amounts of hemoglobin in the extradural CSF, or even into the scalp tissues. So with respect to your sensor, how is your sensor operation and particularly its calibration affected by that?
Thank you, Dave. Very good question. And it's something that we are trying to resolve. Obviously, when we place the sensor on the forehead of these patients, the neurosurgeons very carefully, they are trying to avoid basically, we don't want to put the sensor on a place where there is a hematoma underneath, because obviously we don't know how that will affect the PBG. And this is what we are, as we speak, we are currently trying to understand the behavior of optical signals, in this case PBGs, coming from a part of the brain which is actually affected with a hematoma. It will have the PBGs coming from that area, the absorption of light in that part of the brain, which obviously impacted. It will be very different from a part of the brain that is vascularized in a normal way. We don't know, to be honest. Yeah.
Okay.
And in your question,
I didn't have an answer myself. I knew.
No, I don't either. We are doing some now in the in vitro. We are inducing a hematoma in the in vitro model we did. We developed a phantom, like a adult brain, and we're trying to induce artificially a hematoma, and we see what happens or a hemorrhage into that part of the brain that we are optically interrogating. And it will be very interesting to see what happens to the optical signals. And in relation to your melanin, you're absolutely right, especially when you're looking for chromophores, the absorption of, obviously in the visible range of light with increased melanin, it will have an impact on the signal and coming back or coming through, which it will impact in your measurement. You're absolutely right.
Okay, thanks.
Thank you very much indeed. Nikolai Dimov, you had a question for Tong. Would you like to ask that question, please?
I was muted, sorry. Thank you for the lovely presentation. And I think Professor Tong already answered in the chat, but essentially I was wondering if the SPR technology that was presented was also applied to some biosensing and for instance, can it be combined with antibodies or aptamers and also detect either fluorescence or the SPR, I don't know which effect is more long lasting and sort of in the vicinity of the sensing area. But if you could comment on that, that would be fantastic. Thank you.
Oh, thank you. Yes, the answer is yes. We have explored localized surface plasmon resonance effect, which means we coated the fiber not with gold film, but with gold nanoparticles. That creates similar to the SPR effect, but we call it a localized surface plasmon effect because the coating is not a zinc film, but it's particles. And then the gold nanoparticles is functionized with antibody, and then antibody is able to interact with an antigen. And then their interaction will cause actually, again, dielectric signal changes and cause this localized surface resonance wavelength. The answer is yes. But interestingly, if you are using thin film or you are using nanoparticle coated SPR effect, the sensitivity is slightly different. So we have spent a lot of time. If we're directly using solid gold nanoparticles as a coating material compared to the thick film, thin film itself, thin film sensor is more sensitive than the nanoparticle coated sensors. So we spend a lot of time to improve the sensitivity of localized surface plasmon sensors by changing the nanoparticles in terms of its shape, from sphere to rod, from alloy to even to the hollow structure. We realize actually when we make the nanoparticles to be hollow, the sensitivity is much, much higher, is almost at the level of SPR effect. So yes, the answer is yes, we have done that for the biosensing, which, sorry, actually, because of time limits, I haven't presented today here.
Thank you very much, Tong. Thank you. Next question is from Susan Pierce for Peter.
Can you hear me now? Sorry, I didn't.
Yes.
Yes, sorry, I didn't seem to have any control of my audio. Yes, so consumer wearables, they're driven by sort of the health and fitness market and the worried well and the people who have disposable income and an interest in these things, rather than where we have the greatest clinical need maybe for these longer term monitoring outcomes. So how can we incentivize the producers in that market to move towards measuring other variables and putting effort into those kind of analysis?
Thank you very much, Susan. Yeah, very helpful question. So I would suggest the first source of incentive would be from device users. And I hope that the development of technology to detect, for instance, possible atrial fibrillation in Apple Watches and other devices demonstrates that device users are concerned by monitoring their health, and they would value this additional technology. In due course, if this technology is shown to be clinically helpful, then I hope that incentive will also come from other areas as well, such as healthcare practitioners. And perhaps in different healthcare systems around the globe, there may be the possibility of providing financial incentive, in that potentially there could be exchange of finance between healthcare providers and device companies if the devices are helpful.
Thank you very much, Peter. The next question is from Brian Memmott.
Hi, Professor Sun. I just had a question. So my background is more clinical as well as research, and I'm just wondering if there was any continuous temperature monitoring that could be used for patients, similar to how we have continuous ECG monitoring. It could help then detect any temperature changes, which could be used to diagnose sepsis and COVID, which require quick action to treat. So I'm just wondering if that's a product that is available, and just wondering how comes in a clinical environment in the NHS it isn't being used.
Thank you for your question. Yes, that is yes, because if we use FBG technique, which has shown in the video, that technique is straightforward for continuous temperature monitoring. The temperature range is up to 1000 degrees Celsius. So any temperature within that range, from -200 to 1000 degrees Celsius, then we can use fiber backgrading technique for the continuous temperature monitoring. Of course, there are some other techniques which can also be used for temperature monitoring, but for continuous monitoring is not a big problem.
Thank you very much.
Can I comment briefly on that?
Yes, please do.
Obviously, within the clinical domain, the monitoring of core temperature, it can be done continuously for patients. Primarily, they're probably anesthetized. They have an esophageal thermometer during the op, or they could have a rectal. I think the need is to be able to monitor continuously the core temperature of patients which they're not anesthetized. And I think for me, and aligns with Peter's on wearables, it would be a small transition to be able to utilize the effect of tympanic thermometry, but in a non-handheld approach. So you have a wearable tympanic thermometer that it can go into any awake patient in a ward or in any hospital place to enable this continuous core temperature monitoring. So you're not dependent into an indwelling thermometer or which is not continuous, especially when the patients are awake.
If I may, just to add another point, if we use fiber backgrading techniques, it is not just to be able to monitor a single point. it can monitor literally anywhere you like. You have a temperature profile to be captured, because it can, wherever the fiber goes, the temperature points be mapped around. So you can have the whole picture of the temperature distribution of the area which you want to monitor, rather than just specifically to a point. So it depends on the requirement. Some of the applications that we have used so far is more looking at the area. So you easily identify where the hotspot is, because there are sometimes it could be 100 sensing points or even more than that to be captured simultaneously. Then in that case, you can see the changes or identify the hotspots very easily using grading approach.
Thanks very much, Tong. David, you have a question for Tong, please. David Delpy.
Okay. I think I'm unmuted now. Yeah, Tong, the question was about cross-contamination and sensitivity of your nano imprinting sensor, because as I said when I typed the question out, the heavy metal sensor is remarkably insensitive to the presence of other heavy metals, but the sensing of cocaine is always very difficult because of the unknown type of cross contaminants or cutting materials that people have put into the cocaine. I sit on HOSAC for the Home Office, so I'm aware of this problem. So the question is, how sensitive is that nano imprinting technique to the presence of unknown contaminants?
Yes, you're absolutely right, David. When we did that work, the reason we decided to use molecularly painted polymer is because of its high selectivity. Once actually the sensor was developed, our contact from Home Office, then she to put a series of different drug samples as well as those interference, them together and come to city. And then she actually sat with us for the whole day, because legally we are not allowed to keep all these samples in the laboratory. So he has worked with the lady from the Home Office to do the test and then look at various types of drugs and extras, and including those cutting agents. And then we very happily to conclude that the results obtained are so promising, because it's selected a cocaine strain. The other interference actually make the minimal impact on the detection. Yes, this has been considered when we actually did the work jointly with Home Office.
Okay, thanks.
Thank you very much, Tong. So there don't appear to be any more questions. So what I'd like to do is to thank the speakers for their really exciting talks, and should have prompted a lot of questions from the audience, and thank all those who've asked questions for the participation. I'd like to thank Ahmed Kovacic, who's been working with me to set up this joint venture, and Graham Dransfield, and then John and Matt for setting up the vision today. Also, special thanks to the Dean of the School of Maths, Computer Science and Engineering, Raj Roy, for his support in setting up this joint venture. Our next webinar is cybersecurity in healthcare, and that will be on the 17th of June. It'll be chaired by Professor Raj Rajaran, and we have two exciting speakers, Philip Booth and Burkhard Schaefer. So please register for that as soon as it's advertised, which will be in the next few days. There are a couple more lectures, 16th of September, Artificial Intelligence in Healthcare. And then the final seminar will be the 11th of November, which is Technology Challenges in Healthcare. So I would like to thank you all for joining in, and keep safe. Thank you very much indeed.